We stopped here last time. So we were talking about DNA technology. DNA technology means it is the use of technology to study DNA, to manipulate DNA, to study DNA. People take DNA from one organism, put them inside another organism, and that way they actually get some products made. The example I gave was the example of insulin. So insulin is produced that way. Initially it was isolated from humans, and it was inserted in a bacteria, so which is shown here. So this DNA that was taken from human, it was inserted in a plasmid DNA. Plasmid DNA is extra chromosomal circular DNA found in bacterium or in bacteria. So it was kind of cut with the help of some enzymes, some chemical scissors, and then it was inserted in this plasmid DNA, and then after that it was put inside bacteria. So bacteria multiplied it multiple times, millions and billions of times, and then after it was successful in doing that, it made those proteins, insulin protein, and insulin is still produced the same way. Another example was, the very first product that actually came to the market by using this type of approach was in 1994, long time ago. So it was a tomato which was called so even though they did not know how to spell, so it is as a flavor saver. So it means those were tomatoes that are actually kept fresh on without refrigeration. So if you take a tomato, it rots after a few days. Once you buy it, you have to refrigerate it. If you don't do it, what happens is it starts to ripen. It becomes over-ripened and it starts to rot. This tomato, flavor saver tomato, would not rot. So it would stay actually on the counter in the kitchen without refrigeration for weeks. So people liked it, but people actually hated it because it was made through this DNA technology called biotechnology. Another name of DNA technology is biotechnology. So you probably have heard this term, right? Biotechnology. Biotechnology means use of technology to work with biology, to work with any living systems. Biology is related to living things. So use of any technology like this one, this is one technology, to produce something or do something to the living systems, produce insulin in bacteria. Or another example you might have heard, that really cute. Have you heard about Dolly? Not Dolly Parton, but the other Dolly, another famous Dolly. So there have been actually several famous dollies, and this was a sheep dolly. It was actually
the first animal clone, right?
This was the first cloned animal. This was the first example of a mammal that was cloned. Actually, people cloned other things before that, but Dolly was the first one that was cloned, meaning it was a replica of the mother that was not created through sexual reproduction. She was created actually by using biotechnology, by cloning process. So this is the power of this type of technology. So once the DNA, foreign DNA, is introduced inside the plasmid DNA, it is put inside bacteria, and bacteria multiply much more rapidly. They grow every about 20 minutes. They split every 20 to 30 minutes, so they make more copies of themselves repeatedly. They grow very rapidly, so anything which is introduced in them from outside multiplies very rapidly. And last time we also talked about transgenic. Transgenic, what does transgenic mean? Transgene or transgenic, what does that mean? Transgenic means
Transfer.
that the one that has a transfer DNA. Transgenic means an organism that has multiple types of DNA, that has transfer DNA in it. Transgenic. Trans means transfer, genic means DNA or genes. Something that has some foreign DNA in it. About 40% of our crops, soybean, cotton, and other crops that we consume are made of these type of transgenic systems. Transgenic systems also are called as GMOs. What are GMOs?
Genetically modified organisms.
Genetically modified organisms. So all these crops that we are using nowadays, about 40% of those crops, actually more than 40%, are transgenic now because they have some foreign DNA in them. They are either resistant to insects, or they are resistant to drought, or they are resistant to weedicide, or they are resistant to pesticides or something in them. Or they just have better quality. Like the example we talked about last time, rice. Rice is naturally deficient in some vitamins. What scientists have done is they have isolated that gene from bacteria and introduced in rice. So the rice quality is much better now. Otherwise, the people whose staple is rice, they have some diseases because they eat rice, especially in Asia, where they find a lot of rice there. So people eat rice actually 3 times a day, and if they keep on eating that rice repeatedly for years, then develop some diseases because rice is not a complete diet. So because they don't have other food, so they just keep on eating only one food. So now scientists have improved rice by introducing a gene from bacteria into rice, and now it has become much better. Okay, another biotechnique or biotechnology related technique or DNA technique is called DNA sequencing. So we have heard quite a bit, it has been actually in the news for many years. The way we have heard this in news is Human Genome Project. So Human Genome Project was a project that was aimed at sequencing or finding out the order of DNA. So DNA is made of those 4 nucleotides, adenine, cytosine, guanine, and thymine. So this project was aimed at finding the order of those 4 nucleotides in all the chromosomes. We have 23 pairs of chromosomes, so that was the whole project. It took about 10 years to do that. And it is regarded as actually even a bigger project than landing on the moon. So back in 1969, when the astronauts actually landed on the moon, that Apollo mission landed on the moon, that was regarded as the biggest jump of science ever. So this one is actually regarded almost at the same level, because it's such a big project that was done that it was actually considered impossible at some point, but it was done and completed in 2003, 10 years of that. So based on that, we are able to understand the complete order of all the chromosomes in humans and many other organisms now. So what we see is this type of a sequence. And since we know DNA sequence is equal to genes, genes are the sequences of DNA, so now we know that what type of genes do we have, what type of proteins are made from those genes, with the hope that at some point we will be able to use that sequence to understand how to control diseases. That was the aim of that project. So there were 2 goals. One goal was to find the order of nucleotides, and other goal was to understand diseases and find some control for those diseases. So the second part of that has not been realized yet, but hopefully it will be in future. So sequencing is one of those. Another technique is called PCR, polymerase chain reaction. So polymerase chain reaction is a technique, is a DNA technology, which is designed to mimic DNA replication in the lab. So naturally, when DNA replicates, when the cells divide, before cells divide, DNA is duplicated or replicated. So scientists actually use the same information that they studied from natural DNA replication to mimic and do the same thing in the lab. So, and in this case, in polymerase chain reaction, it is called polymerase chain reaction because it is a chain reaction in which is one double-stranded DNA becomes 2 double-stranded DNA, and then it becomes 4 double-stranded DNA, as if it is a chain, as if it's a kind of a chain reaction that is taking place. Now, so somebody starts this reaction, and then it continues to go through this process. So then this becomes double, this becomes double, this becomes double, double, double. So it is called an exponential increase. Each next stage is twice the number of DNA than the previous one. And it was all found out through studying the natural DNA replication process. So now, what is the importance of this? The importance of this is that it is the most used technique in biology. Most used technique in biology since it was introduced in 1980s. Long time ago, this technique was introduced, and since then, it is being used very, very extensively in lots of different types of investigations. One of the applications of this technique is in forensic science, where a single sperm or hair follicle or some skin cells are found on the crime scene. So what scientists do, when forensic scientists do, they take that DNA, even if it's a single copy of DNA, a single sperm cell or a single hair follicle is enough for that technique. It is so sensitive. So then they take it and run it through this analysis, which is called polymerase chain reaction, and then they are able to find out which of these suspects DNA matches with the DNA on the crime scene. From a single cell, they are able to do that. So what they do is they take some samples from the suspects, and then anything that they can collect from the crime scene, they match those profiles. Now, without really going through all this detail here. So for example, in this case, these are DNA that was found from the crime scene and the DNA that was collected from the suspects. So here you can see actually this amplifications, or this PCR profile matches with this suspect here, because it makes the same type of DNA amplification. So it is very, very sensitive technique that is used for that purpose. And it is also being used for the purpose of diagnostics. A lot of actually tests that are given to in the pathological labs, in the clinical labs, are based on PCR. So when they try to find out what type of virus or what type of bacterium is infecting, so actually they use this technique because this is very sensitive type of a technique. Also in cases like cancers, this technique is used for diagnostic purposes. So this technique of polymerase chain reaction is to increase the number of DNA, the number of copies of DNA. That is the important part of it. And then its applications are in diagnostics and diagnostics of any type, clinical diagnostics, and in forensic science, where things are collected from the crime scene and those are matched with the profiles that are collected from the suspects. And this technique has helped actually a lot of people. As I mentioned last time also, a lot of people who were wrongfully convicted many years ago, back in 1950s and 60s and 70s, if they're still alive. So from 60s and 70s, a lot of people were convicted on some charges, mostly kind of sexual assault type of charges. So they were sent to jail. They actually served like 20 years or 30 years. Based on this PCR technique, they have been exonerated because their profiles did not match. So even though they were sent there for more than 2 decades, there are actually many people who have been, because this technique is so sensitive and very small amount of any evidence can be used for that purpose to match with the suspects. Another technique, another DNA-based technique, DNA technology is in the form of stem cells, even though it is not really directly related to DNA technology, but it is being used to manipulate things. Again, because stem cells, what have you heard about stem cells? They're bad, they're good?
They are really good for helping with cancer victims, like the bone marrow, stuff like that.
Bone marrow transfer, yes, that's right. What else have you heard? That's right. So yeah, there was actually a lot of controversy about embryonic stem cells, which are
Can they keep
taken.
them from the cord now, though?
They now actually you don't even have to have the cord. They have found some stem cells which are present throughout your body. You have so many different types of stem cells. Your eyes have stem cells. Your muscles have stem cells. Your brain has stem cells. So they are everywhere. At that time, embryonic stem cells were the only one. The embryos were the only source of stem cells. So that's why there was a lot of controversy, and people did not want to or scientists to work with the stem cells, because they thought embryos, if they are allowed to continue, they will become babies. Even though that controversy was actually kind of not really understood very well by people, because the thing was, those embryos were taken from in vitro fertilization clinics, where people actually go to get those test tube babies. So, and then all the other embryos that were left there, they were usually discarded. So anything that is left after the procedure is done on a couple, all those embryos were discarded afterwards, and they had no chance of developing new babies from them. But the people actually misunderstood it and made it a big controversy.
And wouldn't be kind of the same thing as just like menstrual cycle, like getting rid of the rest of cycle, anything that's been fertilized?
But the normal ovulation does not result in fertilization.
So I don't think that's a big deal.
That's right.
Mad about it.
Well, I mean, it was not actually because of the politics involved,
Yeah.
So we know what happens when the politicians get involved in these type of things. Sometimes they don't understand all the nitty-gritty of these things, but the only thing that they hear is, oh, embryos, that's it. That was enough for them. After that, they would just shut down. So, but now actually that controversy is not there. Stem cells actually have a lot of potential, and stem cells are now being used in some therapies. They can help in cancers, they can help in some genetic defects, and in some countries, not here in the United States yet, but there are some experimental therapies that are available. Actually, when I used to teach immunology, I had some students at FSU at that time. They had some genetic condition in their family, so and they actually had gone through some stem cell therapy here at Duke University. So what they did was actually they isolated some stem cells from the person, and then they re-injected back inside the body. So if the same person had those stem cells, isolated them, and then grew them in the lab, and then injected them back in the body, and people actually got cured from that genetic condition. Or at least it helped them to some extent. So this is the potential of stem cells, because these are the cells that give rise to all tissues of our body. So in an embryo has stem cells, which are embryonic stem cells, and those few cells give rise to the entire structure of the body. So they know what to do. So a single cell can make pretty much the entire body. A single embryonic stem cell knows what to make and how to make the entire structure of the body. Adult stem cells, which are stem cells found in different tissues of the body, they have limited potential, meaning that they can make only some types of things in the body. Embryonic stem cells can make pretty much anything. So because they are found in the embryos, and embryos make the entire body. So adult stem cells are considered as pluripotent, meaning that they have, those are called actually pluripotent. Pluripotent means they have potential to make only some types of tissues or some types of cells in the body. Embryonic stem cells are called totipotent, meaning total. They make everything. They can make any types of cells in the body. That's why they're called totipotent. Pluripotent means they are plural in form. They can make some or many cells, but not all cells. Embryonic stem cells can make all types of cells of our body. So these, for example, stem cells that are isolated from the bones, they can make different types of things from the bone. Like they can make some blood cells, because blood cells are produced in the bones. They can also make some fat cells, which is also made in the bones, and then they can make the bone cells, different types of bone cells. So they have very limited types of of differentiation or different types of cells that can be made from them. Embryonic stem cells can make neurons and muscle cells and red blood cells and all types of cells in our body. So they are called totipotent. Those ones are called pluripotent. So the controversy we talked about, controversy was mostly related to the ethical issues. Religious people thought that we are trying to play God. Scientists are just taking the things that we should not be interfering. But what scientists were trying to do was really to find out some ways to cure some of the diseases for which there are no cures. Some genetic diseases. So that's what the controversy was spread from those people who did not want scientists to really use those stem cells. How much time do we have? Still a few more minutes. Okay, so the next topic is more related to the cloning process. Cloning process, which is to make copies or make clones. Identical twins, for example, are clones. And it's a natural process in some organisms, like bacteria. They go through cloning process when binder fission, when two bacteria are formed from a single bacterium. Then that is the cloning process. That is a sexual reproduction process. And the same thing takes place in these yeast cells. So they also go through this type of reproduction, which is called budding. There is no sexual reproduction involved here also. So it is done in small organisms like bacteria and protists and yeast cell. And when the same thing was done in case of frogs, people actually made clones of frogs long time ago, back in 1960s or so. And then in 1990s, they started the process of cloning, which was called nuclear transfer. So what they did was, the way Dolly was produced was, they actually took cells from two different sheep. This is one sheep which was considered as the donor of the egg cell. So egg cell was isolated from this mom of Dolly. And then they also took some cells from another sheep. So this cell that took from this sheep, they actually removed, what they did was they removed the nucleus from this cell, and nucleus from here was isolated, and it was inserted inside this cell. So nucleus from here, from the mom, and then the cell that was taken from here, they took the cell from mom, and then they took the nucleus from another sheep, and then they put those two things together. Then they grew that in vitro in the test tube, and then they implanted that embryo. After it was developed in the test tube, they implanted it in a surrogate mom, a third sheep. So this was a female. This one did not really have to be even a female in this case. So since they took it from the female, they took the nucleus from a female. So when the dolly was produced, so dolly was actually produced through a live birth. So it was not produced in the test tube, it was implanted in the womb of the mother, and it was produced, when it was produced, it was a female sheep. So this is the cloning process. And cloning process can be done pretty much with any organism, with any mammal or other types of animals too. People have tried that with many other types of animals now, and they have been very successful. And I would not be surprised that the meat that we are eating is actually produced through this process also. Now, back in about 10 years ago or so, USDA actually kind of gave some of that meat to people to try. They did not tell them that was a cloned meat, so people thought there was no difference. When they told people that this was a cloned meat, they said, no, we don't want to eat that anymore. So, because it's just the concept. We don't really understand that there is no change in the meat that is produced through this process. It is meat, is still meat the same way. It's just the process is not sexual process, so it is okay to eat that meat.
Why would they clone it? Is it cheaper?
It's cheaper, and plus you can control. You don't have to wait for the mating process and complete that, plus the variability. So there is no variation. So like crops, when you see crops, they are identical. So the same thing they're trying to do with the animals too. If they produce a herd of sheep sheep or cows or goats and things like that, they should be very similar. Because they can do a lot of control over those things. All right, we are going to just stop here. Let's see if we can do that.